I'll be strict. Honestly,
you got it.
I promise. Look, I've even started already. See, I'm losing time. Hello. So my name is Jenny Molloy. I work in Sciences, where I coordinate the Synthetic Biology Strategic Research Initiative and OpenPlant. I also work on Content Mine, which Richard mentioned earlier, but that's not what I'm talking about today. So I wanted to briefly describe OpenPlant. So we're a synthetic biology research centre, and we're all about open technologies for synthetic biology. And synthetic biology is essentially taking an engineering approach to biology. It's a fairly broad field, but we want to engineer plants to do useful stuff. And so when I say open technologies, we are interested in making our data available, making our code available, making our publications available, but we're also interested in opening up a far broader range of foundational technologies to ideally enable innovation further up the chain. So we're interested in material transfer agreements where biologists send biological samples between different labs. They're currently quite restrictive, and so we're working with the BioBricks Foundation and Stanford to develop an open material transfer agreement to kind of aid that. We're also working on open hardware, so you've already had one example, and you'll hear another one later on. So whereby the actual tools that we use to do biology and do biotechnology are accessible and are available, are hopefully lower cost, but most importantly are modifiable, so that you can customise them to your own experiment, and you're not spending £50,000 on each experimental rig, which is just not even in Cambridge is that a good thing, whereas elsewhere when you've got lower resources, we just want to make the science, the biotechnology, cutting edge biotechnology more accessible for different groups of people. And it's an experiment, and we're excited to see how this idea of open source, which is actually quite, so synthetic biology came out of a computer science background. It was essentially open source software kind of inspired. How that is going to work when it comes head-on into collision with traditional biotechnology, which is very patent-driven, very hierarchical, very vertically integrated. And so, yeah, we're excited to be part of the experiment. And if anyone's interested in getting involved, we have numerous meetings around Cambridge. So we have one on Monday, in fact, which is going to look at, one of the speakers is looking at a very interesting project called SC 2.0, where they're refactoring the entire yeast genome by splitting it out into different labs. And actually undergraduates are doing a lot of this work, and it's all open. It's openly collaborative, open data. So come along, you can look us up online to that. We have monthly meetings at MakeSpace if anyone's interested in building open hardware for science. And we're looking into building a bio-MakeSpace in Cambridge, where you can not only build your open hardware, but also test it out. So if you're interested in any of those things, come and talk to me. And I'm really excited to be part of the organising committee. And now I need to stop talking. Thank you.